Hello interstellar everyone, welcome to class 12 biology. In our previous video we discussed about DNA, the structure of DNA. We discussed about the double helix model proposed by Watson and Crick, and I also told you that DNA is the genetic material for most of the organisms. How exactly did scientists come to know that DNA is the genetic material and not some other biomolecules? We have other biomolecules as well, like proteins, carbohydrates. Why are those molecules not the genetic material, and why is DNA the genetic material? So our understanding of the biochemical nature of the genetic material came out from many experiments conducted by many different scientists. So we have different scientists like Griffith, Avery, MacLeod, McCarthy, Hershey, and Martha Chase, who contributed through their experiments in our understanding of the biochemical nature of the genetic material. So these days we can simply say that DNA is the genetic material, but how did we come to know that DNA is the genetic material? All the credit goes to all these scientists over here. So we will discuss about their work one by one. So in 1928, Frederick Griffith in 1928 conducted experiments. He was conducting experiments with Streptococcus pneumoniae. Streptococcus pneumoniae is the bacteria which causes pneumonia, some kind of lung infection in which our alveoli they get infected, and it gets filled up with fluid, and we have difficulty breathing. So that particular disease called pneumonia is caused by Streptococcus pneumoniae, and Frederick Griffith was working with that particular bacteria. And when he was working with Streptococcus pneumoniae, when he was growing Streptococcus pneumoniae in petri dish, he found out that there were two strains of the bacteria. One was a rough strain, and another was a smooth strain. So the difference between the rough strain and the smooth strain was the smooth strain, it contained a mucus coat. Right? It contains a mucus coat over it, and that mucus coat is made up of polysaccharides. Whereas the rough strain bacteria colony, it did not have the mucus coat. So there were two strains, one which produced the mucus coat and the one which did not produce the mucus coat, and the one which did not produce the mucus coat was called as the rough strain, and the one which was producing the mucus coat was called the smooth strain. So there were two strains of bacteria. And he found out that the smooth strain bacteria was actually the virulent one. So what do we mean by virulent? So the smooth strain bacteria had the capacity to cause pneumonia, whereas the rough strain was harmless. So harmless and virulent, these are the opposite terms over here, right? How infectious the bacteria is. So the bacteria which causes the disease is the virulent one, which is the smooth strain bacteria, and the one which was harmless, it was the rough strain bacteria. So in his experiment, what he did was he took these two bacteria, rough strain bacteria and the smooth strain bacteria, and he injected the bacteria into the mouse, right? So rough strain bacteria, when he injected it into the mouse, it did not develop any disease and the mouse lived. Whereas the smooth strain bacteria, when it was injected into the mouse, it developed pneumonia and it died. So in his next step, what he did was he heat killed the smooth strain bacteria. He heated the smooth strain bacteria and the smooth strain bacteria died, and he injected the heat killed smooth strain bacteria into the mouse, and he found out that the mouse lived. Right? When he heat killed the smooth strain bacteria, the mouse lived. And then what he did was he took heat killed smooth strain bacteria and he mixed it with live rough strain bacteria. And when he injected that particular mixture into the mouse died. And it not just died, inside the body of this dead mouse, he found out that there were smooth strain bacteria. He found out live smooth strain bacteria inside the dead mouse body. So what did he propose through this one? He proposed that the R strain bacteria was somehow transformed by the heat killed S strain bacteria. the R strain bacteria, when it was mixed with heat-killed S strain bacteria, what did he observe? He found out that in the dead mouse body, he found live S strain bacteria. So he said that the R strain bacteria was somehow getting transformed by the heat-killed S strain bacteria into this virulent form. So he said that the transformation must be due to the transfer of genetic materials. Some kind of genetic material, some genetic material was being passed down from the heat-killed S strain bacteria to the R strain bacteria, so that the R strain bacteria was getting transformed into S strain bacteria. So that was his proposal. That was his conclusion from his experiment. However, in his transformation experiment, he was unable to determine the biochemical nature of the genetic material. He said some genetic material was being transferred from the heat-killed S strain bacteria to the R strain bacteria, but he was unable to tell the biochemical nature of the genetic material. So then in 1933, these three scientists over here, Oswald Avery, Colin MacLeod, and Maclean McCarthy, these three scientists, they replicated Griffiths' experiment in order to determine what is the biochemical nature of the genetic material which transformed the bacteria in Griffiths' experiment. So they were trying to determine whether the genetic material was protein, DNA, or any other biomolecules, or like RNA, lipids, carbohydrates. So there are many biomolecules, right? You must have studied that in your class 11. So they were trying to determine whether the genetic material was one of those biochemicals. So what they did was, in their experiment, in the series of experiment, they took S strain extract, the virulent strain, the heat-killed S strain extract, and they mixed various enzymes in order to purify the extract. So let's see over here. In the first test tube, they added amylase along with the S strain extract. So what that amylase enzyme will do is, it will digest all the polysaccharide present in the S strain extract. So all the polysaccharides will be destroyed over here due to addition of amylase enzyme. So when this particular extract, in which the amylase was added and the polysaccharides were destroyed, when this extract was mixed with R strain bacteria and injected into the mouse, it still died, right? And then they, likewise, they added other enzymes like lipase. Lipase is the enzyme which digests lipids. Then they added RNase. RNase, they digest RNA molecule, ribonucleic acids. So then they added protease. Protease are the enzymes which destroys protein, or the enzymes which digest protein are called as protease. So when amylase, lipase, RNase, and protease, when these enzymes were added into the S strain extract, and when these extracts were mixed with the rough strain bacteria and injected into the mouse still developed pneumonia, and then the mouse died. So all these mice over here, they died due to pneumonia, and in their dead bodies they found live S strain bacteria. So the transformation has occurred even though these molecules, these biomolecules were removed. Polysaccharides, lipids, RNA, proteins, when these biomolecules were removed from the S strain extract, still the rough strain bacteria was transformed into the virulent S strain bacteria. So the transformation still occurred. So they confirmed that polysaccharides, lipids, RNA, and protein, they were not the genetic material. So over here, what they found out was, when DNase was added, when the enzyme DNase was added, what will happen? The DNA molecules will be digested, the DNA molecules will be destroyed, and when this was mixed with R strain bacteria and injected into the mouse lived, right? It did not develop pneumonia, and inside its body there were no live S strain bacteria found. So what did this prove? This proved that it is DNA which is the genetic material. So when the DNA is destroyed in the S strain extract, the R strain bacteria, it was not getting transformed into S strain bacteria. Therefore, the mouse was not developing pneumonia, and the mouse lives. So this experiment seems conclusive, right? This experiment seems conclusive that the DNA is the genetic material, not the other biomolecules. But the scientists were still not convinced by this particular experiment by Avery, McLeod, and McCarthy. Why? Because during their time, during 1933, most of the biologists, they believed that proteins are the genetic material, because protein was playing a lot of different roles in our in living organisms' body, right? There were enzymes, there were hormones, they took a role in formation of cell wall, cell membrane, and many different things. So they felt that proteins, since they seemed involved in so many different bioprocesses, they thought that proteins may be the genetic material, not DNA, RNA, or something like that. So they were still not convinced. So Avery, McLeod, and McCarthy, even though their experiments seemed conclusive, right, the scientists during their time, they were not convinced. So the confirmation that DNA is the genetic material came in 1952 through the experiments conducted by Alfred Hershey and Martha Chase. So what Hershey and Chase, what they were doing was they were working with viruses called as bacteriophages. Bacteriophages are the viruses which can infect bacteria. So let's say we have a bacteria. The bacteria, it gets attacked by bacteriophage, this kind of bacteriophage, and what it will do is it will inject its genetic material within the bacteria, and inside the bacteria it will produce copies of itself. It will produce many viruses within the bacterial cell, and the bacterial cell, after some time, it will burst and the bacteria will die. So that is the bacteriophage. So bacteriophages, they can infect bacteria. So if we look at the structure of a typical bacteriophage, we find that it is made up of protein and DNA. So here, the black portion over here, it represents protein, and here the blue one, it represents the DNA. We can see that inside this particular protein capsule, the head portion, the protein capsule, it carries DNA within it. So this is a bacteriophage structure. You have to remember this structure. So what Alfred Hershey and Martha Chase did in their experiment was they produced two different kinds of bacteriophages. One bacteriophage whose protein codes were radioactively labeled. How did they achieve that? They achieved that by growing this particular bacteriophage in a medium containing radioactive 35S. It is a radioactive form of sulfur. When bacteriophages were grown in medium containing 35S, radioactive sulfur, only the protein codes get radioactively labeled. Why? Because the proteins, they contain sulfur, not the DNA. And they produced the second type of bacteriophage in which the DNA was radioactively labeled. So in this particular virus, the DNA is radioactively labeled with P32, 32P. This is a radioactive form of phosphorus. So when a bacteriophage is grown in a medium containing P32, 32 phosphorus, the radioactive phosphorus, only the DNA gets radioactively labeled, not the protein, because DNA contains phosphorus, whereas protein, it does not contain any phosphorus. So I hope you understood how they were able to achieve the production of these two different forms of bacteriophage. The one in which the protein code was radioactively labeled, the another in which the DNA was radioactively labeled. So let's look at their experiment step by step, right? So in the first case, right, this bacteriophage whose protein code was radioactively labeled, when this bacteriophage was allowed to infect a bacterial cell, when this particular bacteriophage was allowed to infect a bacterial cell, Hershey and Chase, they found that there was no radioactivity within the bacterial cell. There was no radioactivity within the bacterial cell. That was one significant result that they found. And in the second case, in which the bacterial cell was infected with the bacteriophage which contains radioactively labeled DNA, when they observed that particular bacterial cell after the infection, they found radioactivity within the bacterial cell. This proved that DNA is the genetic material, not the protein. Since the protein code which was radioactively labeled was not found within the cell, they were able to tell that the bacteria was not infected by the protein coat but by the DNA. How did they confirm that? They confirmed that by infecting the bacteria with DNA with radioactive labeled DNA. So the radioactivity was found within the bacterial cell, which was infected with bacteriophage, which contained radioactive labeled DNA. So that's how they confirmed that DNA was the genetic material, right? So this particular diagram is given in your textbook. You have to study this particular diagram thoroughly. You have to remember what I told you regarding the experiment conducted by Hershey and Chase. You can draw this simple diagram whenever examiner asks you to explain how Hershey and Chase were able to confirm that DNA was the genetic material and not the protein through their work with bacteriophage. So there are 3 steps: infection, in which the bacteriophage is made to infect the bacterial cell, and then there is blending, in which the protein cover is removed through blending, and then centrifugation, in which the bacterial cell and the remaining ghost particles, we call them as ghost particles, the protein part of the bacteriophage is removed, right? So through centrifugation they are separated, and then they try to observe for radioactivity, right? So radioactivity will be found within the bacterial cell which was infected by bacteriophage, which contained radioactive labeled DNA. So that is the explanation of this particular diagram. So you have to study this particular picture, okay? So in the conclusion of this particular video, I will try to explain the properties of genetic material, right? So we have already confirmed through experiments that DNA is the genetic material. So Alfred Hershey and Martha Chase, they confirmed that DNA is the genetic material. So let's look at the properties of a good genetic material. So an ideal genetic material should be able to replicate. It should be able to produce copies of itself. Second property should be, it should be stable. It should be stable in such a way that it should also provide scope for slow change, right? It should be stable. It should not be very unstable. It should be stable so that genetic information can be carried out from generation to generation within a species, right? So it should be stable enough, but it shouldn't be so stable that it won't allow any kind of change, because changes, mutation, right? The changes in the genetic material called mutation is a good thing because it provides genetic variability. It produces genetic variability, and variations are really important for the evolution of species. So genetic material should be stable, but at the same time it should also provide scope for slow mutations, okay, so that the organism can evolve. And finally, it should express itself in the form of Mendelian characters. So all these characteristics are fulfilled by DNA, right? Even though RNA also can replicate, right? Even though RNA also can replicate, it fulfills this first criteria. However, RNA is less stable than DNA. Remember when we discuss about the nucleotide structures of RNA and DNA, right? RNA is made up of a ribose sugar, that extra OH group at second carbon. It makes RNA very reactive. So RNA is not that stable. Moreover, RNA also acts as catalyst, biocatalyst. So RNA is unstable. It is highly unstable. Therefore, the viruses which contain RNA as a genetic material, they are able to mutate. They are able to mutate at a much faster rate. So RNA, even though it can also act as a genetic material, it is very unstable. So therefore, RNA is not a good genetic material. Therefore, DNA acts as genetic material for most of the organisms. So how can we say that DNA is more stable than RNA? We can look at Griffiths experiment. In Griffiths experiment, the heat killed S strain bacteria, right? The heat killed S strain bacteria. It still contained viable DNA, right? Therefore, the heat killed S strain bacteria was able to transform the R strain bacteria. So the stability of the DNA can be explained by the complementary base pairing between the two strands. When the two strands, they come together, they form they form bonds with each other, right? Adenine binds with adenine binds with thymine, and guanine binds with cytosine, right? So that complementarity, it provides stability to the DNA. When the DNA is heated, the hydrogen bonds will be broken, but when it is cooled down, what happens is the two strands will come together, and it will regain its structure, right? So DNA is much more stable than RNA. And DNA, it undergoes mutation, but at a much slower rate compared to RNA, right? And DNA, it also expresses in the form of Mendelian characters as we know it, right? So in your textbook, it is written that DNA, it is a good genetic material in terms of storing genetic information, whereas RNA is a good genetic material in terms of transmission of genetic information. So we have the comparison between DNA and RNA. However, in your textbook, it is also written that in fact, it was the RNA. There are many proofs that RNA was the first genetic material. DNA just evolved from RNA. So which is the first genetic material in terms of evolution? We say that RNA was the first genetic material. DNA evolved from RNA, right? So that is about the properties of genetic material, and we compared which one is the better genetic material, DNA or the RNA. Even though both of them, they can act as genetic materials, they have their own advantages, and DNA has got more advantages compared to RNA. So therefore, DNA is the genetic material in most of the organisms nowadays. All right, so in the next video, we will discuss about the replication of the DNA, the first property. I told you that genetic material should be able to replicate. It should be able to produce copies of itself. So in the next video, we will talk about DNA replication. We will see how DNA produces copies of itself before cell division. Yeah, thank you. Please read about this one.